Hello everyone, today we are in Odense Hospital and we do urine measurements with our new Raman microscope. Danila,
Hello, I'm Head of Research and Development in Lightnovo.
Alexei, CEO of Lightnovo, and
Thomas Emil Andersen. I'm a professor at this department of clinical microbiology here in Odense, at Odense University Hospital.
Thomas, what is the purpose of our measurements today?
Okay, so what we're doing today is we are trying to see if we can use this Raman instrument from Lightnovo to figure out if a urine sample is infected or not. So typically we spend a lot of time and money on culturing these urine samples on agar plates, just like with this one. So we put down these urine samples on the agar plate, and we look at the results the day after, actually. So because it takes some time for the bacteria to grow up, but in this case here we are developing this new way of analyzing the samples directly on urine, so doing the Raman spectroscopy directly on urine.
Yes, and when we go closer to the actual measurements here, you can see a multi-well plate that we analyze with a 100x microscope objective, and inside we have the Raman spectrometer that Danila, please continue what it actually does right now.
So setup automatically measured 96 wells. In each of wells there is a dry urine sample, so it can in one go measure all 96 samples, and it takes 16 samples from each well. This is a laser spot, and here you see the urine spectrum with distinguishable features of Raman peaks, which are different for every sample depending on bacteria infection.
And then later we will apply machine learning algorithms to differentiate all of those samples. Hopefully cultivation soon would not be as much necessary thanks to this interesting project. Thank you everyone for your attention.